black hair, red hair, no hair. It's all based on the information contained in these chromosomes. Okay, so with that as a little introduction, now let's get an idea just how big the human genome is. Okay, how long would it take you to read the entire genome? So if you're reading that A, A, T, G, G, C, A, A, if you're reading those 4 letters in that sequence, how long would it take you to read the entire human genome if you read 1,000 nucleotides every minute, which would be somewhat equivalent to 1,000 words a minute? How long would it take you? Any guess?
1,000 years.
If you worked 40 hours a week, in 25 years, you'd be half done. So you begin to get a little bit of the idea of the immensity of the genome, and also why computer programmers and bioinformatics people have started working so close together now, because clearly this is way beyond the ability of any one person on a spreadsheet to be able to handle. You've got to have massive computer space and capability then of quickly scanning through all of these letters. And so it really has become a major computer process of dealing with the human genome. Just to assimilate and to do all the comparisons and all the analysis, it's beyond human mortal capability. It takes computer speed and computer capacity now. And of course, the bigger the computers, the better, because you're still talking about billions, or as Carl Sagan would say, billions of these nucleotides. So, human genome. If you recall, when it was first sequenced, the evolutionists were just giddy about how it supposedly showed all this evidence for evolution, all this evidence for common descent, proving that the humans are part of an evolutionary process, that we share all this in common with primates such as chimpanzees and gorillas, and it proves that we're just part of this long evolutionary lineage. And they just go on, and I remember very vividly listening to just popular radio. And there would be these things come on where they'd be interviewing this one particular scientist or another scientist. They'd be talking about, oh yeah, the genome, it proves this and it proves that, and it's just wonderful how it showed an evolution. Do you hear any of that anymore? I don't. I don't hear it anymore. It's kind of amazing how just in less than 10 years, suddenly what was so super and wonderful and just tremendous support for evolution, they don't even hardly want to acknowledge its existence anymore. Oh, I don't know anything about that. Let's look at this fossil here. And suddenly there's this huge change in direction. And it's now the creationists that are saying, let us at that data. Bring up that genome. Let's look at that thing. Let's analyze that thing. Junk DNA. That was, as I've told you, that one scientist that was the head of the genomics program for the Department of Energy. Junk DNA was his Exhibit A. And you've heard other examples where evolutionists pointed to junk DNA as Exhibit A, demonstrating that the human genome was a result of evolutionary progress, evolutionary descent, not special creation. Okay, what do we mean then by junk DNA? What their claim was, what their claim was is that only 2% of the genome actually functioned. So think about that. All that DNA you have in all those chromosomes, the claim was only 2% of it was useful. The rest of it was just junk. Didn't do anything, just sitting there, just junk. And of course then the claim was, well, no creator would put all this junk in there, so where'd it come from? Well, it obviously came from evolutionary processes. That's how it got in there. Certainly didn't come from creation. Okay, so then the idea was the rest was junk. Okay, junk in the genome sequence, the assumption was that the DNA that's in there that's not doing anything is evolutionary leftovers. It's kind of like your analogy of vestigial organs. This is vestigial DNA. You've outgrown it, you don't need it anymore, it's still there. Okay? Clear as I say, contradictory to the creation model. Creator's not going to put junk in there, so the simple fact there's all this junk in there means you weren't created. And that's the short of the challenge or the assumption made. Okay, where the junk DNA supposedly came from is, we'll say, in evolutionary history at this point, you had certain genes that you were using, obviously, at whatever organism you were at as we're moving up the evolutionary ladder. By the time evolution had proceeded to get creatures into the mammal area up here, you didn't need a lot of the genes you were using back here when you were still just a single or just a small multicellular creature. But not all those genes necessarily disappeared. Just because you stop using them doesn't mean they just disappear. They just aren't being used anymore. So it's like that old refrigerator. You don't use it anymore, so you just stick it out in the garage. And what happens to it is it starts decomposing because you're not using it, you're not keeping it up. Well, the junk DNA assumption was the same way. You're not using these genes anymore, but they don't disappear immediately. They still hang around in the DNA, but they're not being used anymore, so they slowly start to decay. They slowly start to degenerate. It doesn't affect the organism any because it doesn't care, because it doesn't use them anymore. But they're still there. And that's the thinking of junk DNA. So the idea is you had some 90% of this huge human genome that was nothing but all of these leftover refrigerators you didn't use anymore, just sitting there decaying away. And again, no creator would do that. So where'd all the junk come from? Okay, I want you to look at this group, this clutch of 6 mice. Notice the different colors. One's much darker, some moderate color, some much lighter color. There's one thing these mice all have in common. They're clones. Now, from what we originally understood about DNA just even 10, 11 years ago, this was impossible because they cannot possibly have the same genetic information and come out looking 6 totally different colors. Couldn't do that. But they do. And this, in fact, is the 6 mice of the first demonstration of, in fact, principles we now know come into play that are telling us, first off, that there's a lot going on above the actual sequence level of the DNA, and